You get me today instead of David. How do you like that? God damn, there's like some fly in here. It won't leave me alone. Shit. Hey everybody, what's up? Anyway, today we're going to talk a little bit about genomic DNA purification. I think we sent out everybody the replacement primers. And has anybody tested any of that stuff out yet? Anybody tested out the new primers and everything? See if it's working. We also included a DNA extraction buffer, which helps purify DNA from plants better. So we'll use that when genotyping plants. We'll talk a little bit about that today. But yeah, just interested how things are going. Has anybody got a functional PCR to work yet? Seen it on a gel. Anybody. PCR is one of those things that can sometimes be like magic. Like, you don't know why it doesn't work. You don't know why it works. Unfortunately. But yeah, a lot of times with PCR, if your PCR isn't functioning, you just have to start varying different variables and see how it works out. Usually the main thing that you'll vary in PCR is the annealing temperature. So the annealing temperature is the temperature at which you allow the primers to anneal to the DNA that they're going to amplify, right? So you heat it up and DNA separates. You cool it down and let it anneal, and then you heat it up a little bit and let the DNA extend. Then the higher the annealing temperature, the more specific you're trying to get, because the amount of energy that's required to bind for the primer to bind the DNA is more, right? Thermal energy. Not really, Dave. You didn't really miss anything. So, like, the annealing temperature is usually the most common thing that you're going to modulate. That and the extension time. But generally, with the extension time, if you stick to, 1 minute per kb, round up, usually just round up, then usually won't have too much trouble with that. You can go too long, so you don't want to, overshoot it by a minute or two. You want to try to keep it, generally as reasonable as possible. But it's the annealing temperature. So if you're having trouble, if your PCR is not working, generally what you want to do is you want to lower your annealing temperature. It's not always the case, but it's a good thing to try. It allows for more promiscuous binding of primer to DNA. It makes it easier for the primer to bind DNA, so you have a higher probability of your PCR working. But if you kind of can see like a smear on your gel, generally that's because you're getting non-specific primer binding, and it's just amplifying a bunch of different pieces because the annealing temperature is too low. So in that case, you generally want to increase the annealing temperature. I generally don't use annealing temperatures higher than like 56 or 57. You generally, I just, I never have to. That's not saying that no one will ever have to, but generally you want to try to keep it between like 52 and 57. If you're going lower than 52, it's probably something else that's wrong. And if if you need to go higher than 57 for specificity, it's probably something else that's wrong. So if you're having trouble getting your PCRs working, that's a good place to start. You know what? Calculating annealing temperatures, Dave, is really fake. There's no real way to calculate annealing temperatures. Now, you can use um, these calculators online, but it literally is all a bunch of bullshit, you know? So, generally, if if you're amplifying something that's pretty pure and you want your product to be pretty pure, I usually do something like, I don't know, 56, 56.5. It doesn't matter what the primers are. I know this sounds ridiculous. If I'm amplifying something that like genomic DNA or something like that, I'll generally do like 52 to 54 based on what kind of genomic DNA it is. Um, but this stuff is not empiric. It's not empirical, right? It's basically just like a guess. All these annealing temperature calculators. They're all just guesses. Um, don't get me wrong. I'm sure they're pretty good guesses, but like, uh, it's not what has worked for me empirically. Not at all. And yeah, so take it however you want. If you want to use annealing temperatures to try to do your PCRs, I'm probably not going to hurt anything, you know? Probably not. um, but, you could get away with not even caring about the annealing temperature. The majority of PCRs I run, I probably use 55. That's probably the standard annealing temperature I use for every PCR is just 55, no matter what the calculated annealing temperature is. And, I have pretty good success with my PCR. So, I don't know, take that however you will. Do whatever you want in this situation, whatever works for you. Here's the thing. Oh, the new primers work. Great. That's great, Dave. Awesome. I'm glad they worked. That's awesome that you got the first step working. Get your first PCR going. Get that shit working. Um, so then you can start moving on to the other organisms. Um, but yeah, you have to understand that like science is mostly about just what works for you, right? Everybody has their own little things that they do, and everybody does things a little bit differently. Um, and so when people give advice about how to do science, like take that with the understanding that the way it might work for you might be completely different, right? Just the environment that you're doing it in, the way you pipette, you know, like it could be so many different things that go into these things that it's just like, just find what works for you and stick with it. Um, that's that's what you should do. And what I say or what David says, generally, that's just our experiences with this stuff or how we were taught, right? Um, but yeah, my whole life, I've never believed in annealing temperature calculations. And everything's worked out pretty okay. So, yeah, there's that. All right. So, if you got the first PCR working and you have an idea of what's going on, then the rest of the PCRs in this class um, are going to involve extracting genomic DNA. So generally when you're doing PCR from plasmids and stuff like that, you're trying to insert you're trying to change the plasmid, you're trying to engineer something new or create something new by amplifying DNA. Um, so it's more genetic engineering, molecular biology than it is genetics. Um, but it's definitely a skill you should learn how to do. But it's probably also the easiest PCR that you'll ever do, because the two things, the primers and the template that you're amplifying, are relatively pure, right? So you don't have to worry about too much other stuff. So it's really nice. Now, um, the once you start getting into, I think our next experiment, let me check. I think it's human DNA amplification. Now, human DNA, uh, you know, I do it the same way every time. I'll be honest with you. And sometimes it, and it only works maybe about 75% of the time. So, take that into account when you do this. And I haven't quite figured out what is the optimal here. Um, I think it might be quantity of DNA or cells, but I don't really know for sure. Um, so now, a lot of different there's a lot of different protocols out there when you look at genomic DNA purification. Generally, I try to keep it as simple as possible. So, if you want to do human DNA purification, um, you can buy kits for this stuff, right? So, it really depends how into this stuff you're getting. If you're trying to use these genetics for like a company or something like that, you probably want to buy a kit or yeah, so whether it's plant or human cells or, whatever, you probably want to buy a kit for the purification. A lot of protocols that you'll read online use a kit or use beads or use something to purify the genomic DNA before you use it. Now, in bacteria and yeast and human cells, I haven't really found any issues about um, not using a kit or not using something special. While with plants, um, I have, and like animal tissue. Um, those usually have been a little bit more difficult in my experience to get a successful PCR from. So it's good to start first with just human. You can get the cells from yourself, and you can do a alkaline lysis, right? So basically, the way that this experiment works with the human cells is you're going to take a cheek swab, stick it in your mouth, and get some cheek cells. Don't worry too much about it getting contaminated or other bullshit or things like that. That shouldn't be too big of a deal. It's 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 not. I haven't really found any issues with anything like that. Um, so like just take that cheek swab, stick it, sterile cheek swab, stick it in your mouth, get some cheek cells, and then just mix it in the NaOH. Now, the swab will take up some of the NaOH, and that's okay. Don't worry too much about that. Just try to get off a bunch of cheek cells, right? Your solution should become a little cloudy, and you should see chunks and stuff floating around in there. Um, you know, uh, uh, that's how it should look. If you don't really see much in there, go back for like a second time, but don't stick the same swab that you stuck in the NaOH in your mouth. Get a new swab. Um, yeah, use a swab and like, uh, you know, get a good amount of cells in there. It should look significantly cloudy, and you should see stuff floating around in there. You actually be I'm usually surprised when I do it. I'm just like, whoa, that's a lot of cells just from my cheek. So after you get the cells in there, what you're going to do is you're going to boil it and to lyse the cells. Now, this is the case with bacteria and yeast. Um, Bacteria and yeast, you generally don't even have to do anything to them. You can literally just take bacteria, mix it in your PCR tube, and run a PCR, and it should work. I usually like to set the initial denature step. So usually you have an initial denature step for like a minute before you start going into the cycles. Um, I like to set the initial denature step at for like 5 or 10 minutes. Then that allows the sample to kind of like boil, right? So it helps lyse the bacteria, helps lyse the yeast, and you know, when they lyse, they release the DNA, their genomic DNA. Now, the good thing about bacteria and yeast is generally they don't have anything that inhibits PCR. So, kind of the more the better. In my experience, again, this is just my experience. Um, I've added a lot of bacteria and a lot, and I've never seen it inhibit any of my PCRs. Well, how much is a lot? Like more than a colony of bacteria is worth. Same with yeast. You can add a good amount to where you can physically see it, and you won't negatively affect the PCR with bacteria and yeast. Now, human cells, you can't add them directly to the PCR. I've tried. Um, if you can get it to work, let me know. If that's something that you're just trying to wing, let me know. I would be interested in finding uh, a simpler method to just genotype human beings from cheek swabs. But generally, what you do is you add the cells to sodium hydroxide. You heat them for, um, like 95°C for 10 minutes. Um, and it doesn't have to be exact on this. Sometimes what I've done, what you could do is you could take your tube, your microcentrifuge tube, and just stick it in the PCR, um, and set it for 95°C for 10 minutes. Generally, that works just fine. Even if the tube doesn't go all the way in the machine, it'll generally get to a high enough temperature for those 10 minutes that it will lyse the cells. The other thing I do is I just like take a cup or a beaker or something, add water to it, heat it up in the microwave till it's boiling, put a piece of tin foil over it, and then just poke the tube through the tin foil so its lid is sitting on top of the tin foil, and the part with the cells and the NaOH is in the water. And then just let it sit for 10 minutes. This isn't like an exact science, Like nobody here is trying to do this exactly. Uh, so you got a lot of wiggle room with this in terms of temperature and time. It's generally always better to go hotter for longer, but you don't want to go too hot for too long, because what happens then is you will start to get like DNA being cleaved and breaking apart and hydrolyzing. So, you know, don't don't do it for too long, but you also don't have to be like super specific about this heating step. Um, at least in my experience. Now, after that, what you're going to do is the sodium hydroxide is very, very basic, right? So you're going to want to neutralize that. Um, so David does electric kettle. Yeah, exactly. Like whatever you have works. It doesn't. This isn't like a specific temperature thing. So like after, what you're going to want to do is you're going to want to like buffer the pH, because extremely high pHs and extremely low pHs, um, proteins don't like them, enzymes don't like them, DNA doesn't like them. So if you're trying to run like a PCR reaction and you have these really high pHs, it's generally not going to be good for it. Yeah. Even though Taq polymerase is pretty thermostable, I bet you at like a pH of 14, it falls apart. But who knows? I don't know if anybody's checked that or studied that. Um, so yeah, you add the tris, help neutralize the pH, so then you can add the cells to the PCR reaction. Now, in this case, I've found 10 microliters generally works. If you're having trouble amplifying this, what you can do is you can increase the two things on this. If you're having trouble working, is you can increase the amount of cheek cell solution that you add to your PCR. Um, the other is, um, well, generally, so normal PCR, when you're using a really good template, people generally say run around 30 cycles. Um, when you're using genomic DNA from like human cells or bacteria or yeast, generally I like to jack that up to like 35 or 40 cycles. It it uh it works. Sometimes you need that many cycles. So the number of cycles you need is related to your primer concentration and your template concentration, or your DNA, or genomic DNA concentration. So if the primers don't get access to a lot of DNA, or there's not a lot of DNA in the solution, then your amplification is going to take more cycles, right? It's just the way it works. Um, if you start out with 4 copies as opposed to 2, right, you're going to be a cycle behind. So I generally like to run 40 cycles. If I do not, if my template is not purified, I just generally just run 40 cycles. Unless you're in a rush for time. If you're in an extreme rush for time, bump it down to 35. But generally, if you're just setting it and forgetting it, what you can do is you can just do 40 cycles. It doesn't hurt. You're just going to get more DNA. It's all good. So, yeah, do 40 cycles for something like that. Generally, Dave Dave asked, "Do you put the tube in the kettle and boil the water?" David Ishii said that he, boils his samples in an electric kettle. Generally, you want something for the tube to float on. Number one, if you boil for too long, what could happen is the pressure can build up and the tube lid can pop open. And it's definitely happened to me a number of times. And then if it's like just laying in the water, it gets mixed with the water and you lose your sample. Not the end of the world. You can just start over sometimes. But generally, what you want to do is just find something that can float so it's upright. Tin foil works great usually. Um, I'm sure you could find other stuff, styrofoam, um, you know, whatever, just to try to keep it upright. So that if it lid pops open or something like that, um, you don't get you don't get any water in it. Now, I'm sure in a pinch you could just throw the tube in there and say a few Hail Marys and get away with it. But, you know, it's not too difficult to find something that the tube can float with. Yeah, not too difficult at all. So, any questions about the human genomic DNA purification? It's pretty simple, um, though I've definitely messed it up. Uh, don't forget to add the tris, right? That's a good place to just mess stuff up if you forget to add the tris, and you just add the NaOH directly to it. But it shouldn't be too complicated for doing this human genomic DNA. Um, Now, understand that your band may not be as bright as it was for the GFP PCR. The GFP PCR, because we had a good concentration of plasmid, a good concentration of primers, you get a nice bright band. So sometimes bands from genomic DNA are less bright. So just take that into account. And look closely. Um, if you're gonna do this stuff all the time, I would suggest investing in a nice blue light, um, a brighter blue, really bright blue light. You can find blue light flashlights on Amazon for a couple bucks, $3 or something like that work great. Uh, so yeah, if you're having trouble seeing it. So if you're having trouble seeing stuff, generally you can also add more. You can load more in the gel. Um, so you can load as much as you can fit into the well. Also, uh, you can add more DNA stain to the gel. Now, there's like you, after a certain amount of DNA stain, you don't get any return from it. And what I do if I want it to be bright, generally instead of like 5 microliters for 50 ml of gel, I add like 7. Um, you know, if you start getting up to 10, you really don't get much return from that, because the DNA can only bind so much of the DNA stain, right? You want to make it so that the DNA can bind as much as it can and not anymore. So generally, I've found that to be the highest about 7 microliters per 50 ml. But if it's really bright, if you have nice bright bands, you can get away with 5 or sometimes even less. Um, you know, it's what you're comfortable with. What you want to do, how much you want to save of your reagents. Now, plant DNA extraction. So, plant DNA extraction is always a bitch for me. Um, it's and that's because plant DNA extraction, um, it has plants have PCR inhibitors inside their cells, and so it makes it harder to get the PCR to work. Um, Also, plant cells are really robust, right? And generally, you need help breaking apart the cell wall and getting stuff out of it. So, um, yeah. So the DNA extraction buffer um, helps a lot. So the DNA extraction buffer I don't think we wrote what's in the DNA extraction buffer. So let me write it in here. DNA, oh, why is that red? That's weird. Let's make that black. So DNA extraction buffer is made of 1%. It's still red. What's going on? Let's get rid of this red. It's made of 1% Tween 20, 0.5% Triton X100, 150 millimolar sodium chloride, and 50 millimolar tris. And it's approximately pH 8. So, Tween and Triton. Now, Tween and Triton are, I know they sound funny and are weird names. They sound like, like serious chemicals that you'd find in like some sci-fi movie or something. Tween and Triton are called surfactants. Now, a surfactant is something that helps dissolve things that are hydrophobic, right? So generally, when you're using Tween and Triton, what you're trying to do is you're trying to break down the cell wall of a cell. So what these do is they interact. It's like they're basically like a soap, right? So when you get your hands all greasy and you just try to use water to remove the grease or the oil, it doesn't come off. But when you use soap, it comes off. So soap generally has something called sodium dodecyl sulfate, SDS. It's used in science a fair bit, actually. And you can tell a surfactant or, one of these agents because when you shake it, you get bubbles. You get like a lot of bubbles. It actually looks and kind of acts like a soap. So what these things do is they help dissolve the membrane of the cell, right? Because they're like the soap to the lipids in the membrane, right? So cells have lipids in their membrane, and lipids are like oils, right? So these things help break it open because plant cells are they're pretty strong. They don't like to release their DNA, not at all. And these things help that. Now, the other problem I've found, like David said, there's things in plants that inhibit PCR, and this makes it really difficult. So, what you're trying to do is you're trying to add a lot less of the extraction from a plant extraction than you would with like a human cell or bacteria or yeast. So generally what I do is I take 1 ml of the DNA extraction buffer, add it to mortar and pestle, take a leaf, and it's not a bad idea to clean the leaf with soap and water beforehand. Try to rinse it off so there's no soap left before you start grinding it. But yeah, I think we sent everybody a mortar and pestle for this class. If we didn't, then just what you can do is you can chop it up with scissors till it's really tiny. Stab it a lot, scrape it, poke it, do whatever. What you want is you want the DNA extraction buffer to start to turn green. When it starts to turn green, it's because the plant cells are starting to break open. Chlorophyll and other things are getting released into the solution. And so the plant cells are starting to break open. Unfortunately, it's also releasing this stuff that can inhibit the PCR. So what I found works best is you go for a minimal amount of plant DNA extraction buffer instead of more like you use in the other experiments. So taking like 1 microliter and then diluting it down 1 to 10, and then running a PCR, I've been successful. Now, if it's not working, you can go up and down around that number. So if you do 1 to 10 and it's not working, what you can try to do is you can do 1 to 20 and 1 to 5, and see if either of those work, right? Generally, it's a good thing to do. If you're running a PCR for the first time, it's generally good to do a couple different trials with a couple things slightly changed, to, so then it works on the first time. You don't have to set up the PCR 3 times to get it to work. Even if you have a protocol, I just like to vary, something. It's always good to do things twice, right? If you accidentally, like even myself, I generally, when I do any sort of experiment, I do it twice. So I set up 2 replicates or more of the experiment, because you make simple mistakes sometimes that you don't even realize. You accidentally didn't pipette. There's nothing in your pipette tip when you sucked up the liquid, or there's something that you missed or something that you forgot, right? So doing multiple different replicates and varying something in there is never going to hurt anything, right? And usually the cost of this stuff is minimal. So it doesn't really hurt anything, right? So that's what you are going to vary mostly for the plant experiment, right? So again, when you're doing genomic DNA, what I like to do is run it for 40 steps. It doesn't hurt, and it takes longer for sure, but it also doesn't hurt. So run it for 40 steps, and hopefully you get something amplified, right? And David stabs his a lot with a toothpick. Yeah, so those are going to be the major types of genomic DNA extractions that you're going to do with this class. For other things, what I would suggest doing is looking for how people do it in scientific papers, or looking for guides or protocols online. A lot of times there's like a complicated way to do it and a simple way to do it. So don't necessarily use the first thing you find when you're doing a genomic DNA extraction. Look around for a little bit and try to look for something that is a little bit easier, that has, chemicals that generally a scientist would have. Usually these are like tris, sodium chloride, things like that. Tween 20 and Triton X100, those necessarily aren't super common, but they are fairly common on people who do biochemistry and stuff like that, right? But generally, people have done PCR on everything to amplify the DNA of everything, right? So you'll be able to find some protocol out there for the organism that you want to amplify the DNA of. This week, you'll focus on your own samples from your own human tissue. So let's get that to work first. Um, Next week, I think what we'll probably talk about is um, DNA sequencing and how to send your samples off for DNA sequencing. So if you saved your PCR products from your um, from your genotyping experiments in the future, I don't think the human genotyping experiment you can send off for sequencing, but the plants one, the fish and dogs, those you can send off for sequencing. Also, well, actually, you could send the human one off for sequencing. So if you want to send your samples off for sequencing, oh, we'll talk about it next week. I'm not going to go through it all now. But yeah, we'll talk about that so you can get the actual DNA sequence back. That's what's pretty powerful about PCR is, you know, you can amplify a region of DNA that you want to get sequenced and send it off to a company, and for like $6, you can get it sequenced, right? Um, which is really nice. So PCR is really helpful with that. Um, yeah. So, do we have any questions? Any questions about DNA extraction or anything like that? Um, Anything on your mind? As always, like I say, if you have questions while you're doing the experiment, feel free to post in the Google Classroom. If I see somebody ask a question, I usually try to answer it within a half hour or an hour. Obviously, it's not always going to happen if I'm busy or sleeping or something like I'm not going to be able to answer your question, but I try to get to it, in a reasonable amount of time. So don't be afraid to post questions in the Google Classroom. Um, or if you need something like immediate, to find me on Facebook and ask me the question on Facebook, or David the question on Facebook. Um, you know, I have no problem answering it if I'm not busy. So, all right. Any questions? Any questions? You know, alrighty. Thanks for being patient with us. Um, with sending out the wrong primers, and I hope you all get that working. If you start to run out of supplies, just let us know, and we could probably send you more for the cost of shipping or something. So, otherwise, we'll be back next week, same time, and hope to see you all then.